Good morning children, I am Sarida. Now I am going to teach you vegetative propagation. As I told you, vegetative propagation is a method of asexual reproduction seen in plants. And this method is, the plants can produce their offsprings or young ones from the vegetative parts such as stem, root, leaves, etc. And this vegetative propagation is commonly seen in plants which lose their ability to produce seeds. So vegetative propagation is of two types. They are natural vegetative propagation, artificial vegetative propagation. That means the vegetative propagation or formation of a new plant occur in two ways. Naturally they can produce the offspring, and artificially or manually also we can do this vegetative propagation. First we can see natural method. So from the root, stem, etc. some plants are forming their offspring. For example, sweet potato. Sweet potato, we can write it as example, that's better. Okay, sweet potato, then ginger, guava, etc. Here the sweet potato is grown from the tubers of the parent plant, and the ginger, it's an underground stem. So from the underground stem they can rise or they can multiply into new plant, and guava from the buds of the parent plant they can produce the offsprings. And naturally in bryophylla, you already learned in lower classes, in bryophylla plants the offsprings are developed from the leaves. So the leaves consist of glandlets or bud-like structures, and when it lands on soil, it grows into a new bryophyllum plant. It is only a natural method of vegetative propagation. So naturally also the plant can produce their young ones from the vegetative parts such as stem, root, leaves, etc. And these are the examples for natural vegetative propagation. And next we can see artificial vegetative propagation can be done by cutting, layering and grafting. So these are the different methods of artificial vegetative propagation, and you already learned these three types of artificial method in lower classes. In this class you have to learn a new artificial method of vegetative propagation, tissue culture. So along with that we can see tissue culture. Tissue culture. Okay, let's see about tissue culture. Here, tissue culture. Tissue culture is a method of vegetative propagation in which a small piece of the plant, a small piece can be taken from the root tip or shoot tip or even from a leaf also. We can take a small piece of tissue and then it grown into a culture medium or nutrient medium, and we can see the process. So first we can see, we can select or we can take a small piece of tissue from a growing plant. Then it can grow into a culture medium or nutrient medium, and that medium is called agar jelly-like medium, which consists of all the nutrients essential for the proper growth and development of the plant. So that nutrient solution is called agar. So we can put this small piece of the plant part in agar medium. So then it grows as an undifferentiated mass of cells. Undifferentiated mass of cells called callus, that I will show you. So we can select the cut piece from the here, the cut piece of stem we can see. Then it put it in nutrient solution. Then it grows as an undifferentiated mass of cells. So this is the irregular, unorganized and undifferentiated mass of actively dividing cells are formed in this nutrient solution, and this is called a callus. We can't differentiate like a leaf, stem, root, etc. So this is called an undifferentiated mass of cells. Okay, and now we can see, so this callus develop into plantlets. So this develop into plantlets, and each plantlets can transplant into soil or in a pot. So we can raise unlimited number of plants, or large number of plants from a single parent by this method. So this is called a tissue culture. So this tissue culture, within a short period of time, we can raise unlimited number of offsprings or plants from a single parent, from a single part of the parent also. So here, this tissue culture is also known as micro-propagation. Micro-propagation. Is it clear about tissue culture or micro-propagation? Okay. And now we can see the advantages of vegetative propagation. This is very, very important point. What is the significance of vegetative propagation, or what are the advantages of vegetative propagation? You can see, first point, new plants produced by vegetative propagation are exactly like the parent plants. That means they are identical to the parent plant. They are genetically identical to the parent plant. And second one, the fruit trees bear fruits much earlier. So the fruit trees raised by vegetative propagation start to bear fruits than the plant which are produced fruits from seeds. So they can start to bear fruits much earlier. Okay. And third one, the plant needs only less attention than the plants grown from seeds. Then fourth point, many plants can rise from just one parent within short period of time. And the last point, we can rise seedless plants also by vegetative propagation. Hope you understood everything about vegetative propagation, especially tissue culture, and the advantages of vegetative propagation also. That's all for today.